So, welcome back to this course, Functional Genomics. So, in this lecture we are going to look into some of the applications of next generation sequencing. So, in the previous lecture we have seen the principle behind the sequencing and a few examples like cancer genomics. So, we are going to go beyond cancer and look at what are the other applications one could think of using this powerful tool of next generation sequencing. So, here is a paper, it is again a landmark paper which sort of established the power of next generation sequencing. Here they have developed or they have shown the sensitivity at which the sequencing can help as a screen for prenatal measurement of fetal genome. So, you must have read about disorders that affect the children. For example, mental retardation is one such disorder. There could be many other disorders like Rett syndrome and others. It happens because of a de novo mutation, meaning the gamete that led to the embryo had a mutation. As a result, the baby is having a mutation. This is unexpected because the parents are normal, but the baby could be. And likewise in Down syndrome, as the age of the mother pregnant mother increases, the chances of that developing embryo might have Down syndrome increases. So, the traditional method is to look at the amniotic fluid from the uterus and go for screening, either it is a chromosome or DNA sequence analysis and so on. But that also puts the baby at risk because the procedure involved requires invasive procedure. So, if you can come up with certain non-invasive screening methods to understand the genome of the growing embryo, that is going to really help. And with the cost of such sequencing coming down, it can be a routine screening for the developing embryo to see whether the embryo has any genetic defect before a certain developmental stage where the parents could take a decision as to whether they would like to have the baby or not, which legally allowed. So, this paper talks about the non-invasive prenatal measurement of fetal genome. What is that? So, what is known is that for long it has been suspected that some of the DNA or RNA of the fetus, the growing embryo, can be found in the circulating blood of the mother. So, it can come, even few cells can come because the umbilical cord where the exchange of blood takes place, there could be few cells coming into, or it could be only the DNA coming, only the RNA coming, because there are process by which there is now we know that RNA are put into small vesicles, they are transported and so on. So, what they have done is that they have used a method, that is they took the maternal peripheral blood and then captured the DNA that are present in the free DNA that are present in the plasma, the liquid part of that blood, and they have done exome sequencing. Okay? And likewise they have done a shotgun sequencing for the plasma free DNA and then looked at the sequence. So, what does it help? So, the idea is that if you have if the blood carries some RNA or DNA from the fetus, the growing embryo, then by doing this sequencing where you are sequencing every fragment that is captured, and it is so sensitive because these are clonal, meaning that each cluster represents one copy of the DNA, you are able to sequence each cluster, get the sequence separately. You may find that if you know that the mother carries the genome from his father, her father and her mother. So, that is one, and of course, this is a complementary what is shown here, but you can really type that is what is shown here. So, this is the mother's DNA which is representing her, for example, father. This is the mother's DNA that represents her mother, and these are the sequences which are different between father and mother. These are polymorphic site which you are able to genotype and call from whom it has come from. and obviously the fetus would have DNA sequence that had come from her father, right. So, one that has come from mother, the other copy that had come from father. So, this is the information that you have. You have the DNA sequence of the father, you have the DNA sequence of the mother, which tells you what are the alleles that they have, which has come from his father and his mother and so on. And then you have fragments of the DNA that are sequenced using this approach, and all you have to look for is that are there any genome that are representing the signature of father, which clearly tells that yes, that there are genome that represent the father, that certainly should have been from the fetus because it's coming out. So that it tells that there are circulating free DNA derived from the fetus. So that is the approach that they use, and they are able to really show that you can do genotyping of the fetus by looking at the free DNA that is there in the serum, right. And this is a powerful tool because you can type, you can look at the mutations, and you can look at the copy number and say whether it is a Down syndrome and so on. So that's really going to change. Even if it's some more improvement in the technique, it can become a routine screen in the future. It's a possibility. So that's a huge advancement. The other application that can that is of great interest is the 1000 genome project. Here the idea is to sequence the complete genome of 1000 individuals and to look for variations, something that we have discussed about. And these are individuals that are not restricted to a particular race. They have selected from different continents like what is shown here, and they are trying to compare and look at what variations exist amongst the population. The idea is to create what is called the reference sequence. Say suppose my genome sequence has been sequenced. Now when this sequence is done, I need to compare the sequence with some reference sequences to infer what is the variation, whether the variation is something unique to me or present in the population. So for this to happen, you should have enough number of samples already done and present in the database. So that is the objective of the project. So you have at least 100 plus individuals' DNA is sequenced and kept in a database representing each population. So as and when such kind of technology is available for individuals, then they are sequenced, and then you can immediately match with the reference sequence and say whether the variant that was found is common or not common, whether which segment of the genome it represents, whether it could be deleterious, and such kind of analysis can be done using the reference project. So these are the images representing the next generation sequencers. You can see they become very small. Tabletop models have come in. There are different companies that are marketing these products, and they have the chemistry and as well as the the kits for preparing the DNA and ligating into the adapter and so on. So these are some of the popular models that you have. This is just to show you how things have changed. So we started with our discussion on the manual sequencing, how the X-ray film was used to understand the sequences, to the high throughput capillary sequencers. Now what we are talking about is the next generation sequencing, and what we are looking at very soon is a product called MinION. This is all that you have. It's a very small device that you can keep in your palm, which is a personal sequencer. So it's just like your cell phone with a USB card, you can connect to the computer and spot a little bit of your cell or blood or whatever, and it would complete your genome sequencing in few hours. That's what we are expecting. And just to talk about that, we have a video, and I'll play that in the next slide so that you can understand.
Oxford Nanopore Technologies is developing nanopore sensing technologies for the analysis of biological molecules. In Oxford Nanopore systems, the nanopore is inserted into an electrically resistant membrane created from synthetic polymers. A potential is applied across the membrane, resulting in a current flowing only through the aperture of the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. This is the nanopore signal. By measuring that disruption, the molecule can be identified. The MinION is a small portable device for the analysis of single molecules such as DNA, RNA, and proteins. It has been designed for users who want a simple, fast, plug-and-play device that can generate real-time data not confined to a laboratory. Inside the MinION device is a flow cell. This includes the sensor array, which is a collection of electrodes and micro-supports, each one of which has an individually addressable electronic channel. Nanopores are built into membranes lying across the micro-supports. And it is here that the single molecules are analysed, using the change in current passing through the nanopore. The MinION has a simple and fast workflow, and its portability and the versatility of experimental design offers opportunities in a number of different applications. Firstly, the MinION is connected to a laptop through the USB 3 cable. The user then launches the instrument control software. After performing a calibration and quality control procedure, the device is ready for use. The sample is added to a port in the MinION, and fluid flows across the surface of the sensor array, and the user begins the experiment. The nanopore signal is measured by an application-specific integrated circuit in the flow cell and processed by MinO, the instrument control software. This software carries out several core data tasks and can be used to change experimental workflows or parameters. The user receives real-time feedback of key metrics, such as sample quality and number of events. Combined with real-time cloud-based analyses from Metrocore or the user's own systems, analyses can start immediately and continue the experiment until the point where sufficient data has been generated to answer the biological question. The system may be active for minutes or days, rather than being controlled by an arbitrary runtime and subsequent analyses. This is known as run until.
On the bioinformatics side, the systems produce full-length, full-read data, whether it's DNA, RNA, or protein, in real time. And that means you can treat those data bioinformatically in real time as the machine runs. Now, running bioinformatics in real time has a number of advantages. The obvious one is it reduces the compute cost and the management overhead, but it also means you can look for things in real time. If you can see things in real time, you can then feed that back to the system, and you can move the whole system or part of it onto another sample or another experiment.
Simple to use, with minimal sample prep, the MinION is the world's first portable device for the analysis of biological molecules. For more information, please visit nanoporetech.com.
How this device can work. You have to understand all these are commercial things. They are not going to disclose everything that led to the development of this device, but it would give an overview as to what is the principle behind that. And one can buy this. It's not going to be expensive. And if it becomes really, because this has come like a cell phone, and this, you can expect that this would flood. If it is successful, it will flood the market, and sequencing would become just like the blood pressure, or just like people measure the glucose level in your blood. It's going to be that easy. So that's the future, right? So having seen the video, now let's look into some of the advantages and disadvantages of the next generation sequencing. So as is the case with any advancement, any technique or any device, there are something good, there are something not so good. The advantage is, of course, is very high throughput. The number of sequence reaction it can do is enormous. Just put the entire genome, it is able to sequence, and you can scale up, you can multiply, whatever. Very cheap, the data production, but it has its own disadvantage. The disadvantage is it's relatively short reads. So 50 bases, 100 bases, that's all it can read. But what you do is, for the same segment, there are multiple fragments, overlapping fragments you are reading. Therefore, you have to have more reads for a given segment to match, assemble and get the sequence. And because of this process, there is also relatively higher error rates, because there are a lot of amplification process. So every cluster that we spoke about, that video we have shown, that one DNA molecule is stuck to the adapter, then you have PCR-like amplification. The DNA polymerase goes over to make multiple copies, and then that copies are sequenced. So when you are using these kind of polymerases, they are not unlike our cells where the polymerase copies, then there is a mechanism that looks at whether the copying process is accurate. Is there any mismatch? So that doesn't happen here. Therefore, there could be errors. It's pretty high as compared to the traditional Sanger method. And the third is that the data that it generates is so huge that it really requires a very good bioinformatic tool to assemble the sequences to analyze. It's much more challenging. So in a traditional Sanger method, anybody should be able to analyze the DNA sequence, but for a NGS platform, the sequence that it generates requires really skilled person analyzing the data. So that's one of the cartoons that talks about NGS machine, that it's like it dumps, huge data. Sometimes you wonder what you do with that, unless you are able to mine and get the one that you want, and others, what is the noise, what is the signal. So that is something it comes out. So there are many vocabulary people use, some of them in the next generation sequencing. So we'll talk about some. For example, the read, they talk about how many reads. This is a small segment of the DNA, how many times that has been sequenced in independent unit, independent clusters. So the continuous sequence produced by the sequencer, right? Coverage, this is what we spoke about, how many short reads of overlapping segments, that are present. Consensus sequence, so when you have several, sequences, you assemble to get, consensus sequence. And this is a contig. Contig is nothing but you have assembled a small segment, little larger than the reads, but they are not connecting to something else, but they represent much larger as compared to the reads. And then you have to align with the known sequence to get the region, for example, whether it is DNA coding region and all other things. Right? These are some of these issues that come. So we are also talking about single versus paired end sequencing, directional, unidirectional libraries. We will discuss it in a little later. So this is what it is. So you have the reads, these are reads that represent small segments, multiple of them. You got the sequence, and then each sequence is aligned with overlapping sequence to form what is called as a consensus sequence that represent the contig. But still the contig, there are gaps within that. But you are able to say that this contig is located here in the same physical orientation, that you are able to do by aligning the sequence or comparing it with the genome sequence that is available. So this is how you are able to map, right? So this is the workflow, the data that is generated, how do you really study that. So the analysis of the data, basically this is image analysis, because every time a base is added in a cluster, it's going to emit a particular light. So, but in a chip, you are going to have hundreds and thousands of spots for every read. So there's a powerful image analysis tool that calls for every cluster as to what base has been apted. The way you are able to put together the sequence, there are two different methods. One is called the de novo genome, that is, you are sequencing a genome and the sequence is not known previously. Okay? So the way you analyze the sequence is very different as comparing to a genome which has already been sequenced, but you are looking at what difference you have it in this particular genome. For example, human genome is available, I want to sequence my genome and identify what variants I call. So I am going to compare with some existing reference data. So the way you analyze is different. De novo meaning you are generating for the first time. So you have, of course, the DNA fragment, and then you have sequenced it and you are finding these overlapping segments, and the overlapping segments, to make contig. And the contigs are looked at for the sequences, and then you make much larger scaffold like what we discussed. Obviously, this method is not going to give you the continuous DNA sequence. There will be gaps, and one has to fill the gaps with library and other methods, so that is for sure. But in the second method, where you are talking about analyzing the sequence, for example, you want to look into the RNA sequence, or you want to find where a transcription factor binds, or polymorphism or mutations, then basically you are reading single end reads. So one end of the DNA you are sequencing, and then whatever sequence that come, you are comparing with the reference sequence, and that is where you are compiling as to whether sequences are identical or there any difference. The other method is you have a longer DNA fragments. As I told you, the reads are, the length that you can sequence is restricted here, 50 bases, 100 bases, 150 bases, or whatever. So you are not going to sequence the entire DNA fragment, but you are sequencing either end of the fragments, and you are going to, align them like this. That gives you this called as a paired end reads, but in this you are going to have segments that are not sequenced, right. So that is a limitation, but this is good enough for you to tell if you are sequencing this as an RNA, cDNA rather. It only tells how many copies of the RNA is present. So still it gives you information. Likewise, you are talking about chip sequencing data. What are the DNA elements on which your transcription factor bound? So how many times it is bound? So you are talking about the quantitative measurements. These parent reads are good enough because it tells you how many counts that are present. So that's about the next generation sequence and sequence analysis to some extent. Now we are going to talk about much larger application beyond the mutation detection. Of course, one is whole genome sequence, resequencing like what we have seen 1000 genome project and so on. We also looked at, for example, transcriptome analysis, and of course we can try to understand how the genes are regulated in terms of changes in the DNA, in terms of transcription factor binding to that, and epigenetic changes, for example methylation of the DNA, or metagenomics we discussed, where we are looking into a large number of microbes we are sequencing, classifying them based on certain signatures, or paleogenomics, for example extinct species, you can sequence it quickly and understand the sequence and so on. So these are examples. One I am going to show you is what is called the RNA-seq, popularly known as for sequencing RNA, right. So let us say you are looking at two conditions. Condition 1 is a tumor, condition 2 is a normal skin. Let us say the skin with a tumor and skin that is not having a tumor. You isolate RNA exactly the same way like traditional method, and then you are going to pull out all the mRNA if you are looking into the coding RNAs, and then obviously the first step would be to convert the RNA into a DNA, what you call as cDNA fragment, and make it double stranded. And then just like the way you have done it for DNA, you add adapters on either end of the DNA. This is what you do it, right. And then the application is exactly the same like DNA sequencing. You put through the small channels and then allow the DNA to go and bind and make clusters, and then use one of the chemistry to read the sequence. That is what is shown here as a dot, like the way it is explained in the YouTube. And then you are going to analyze the sequence. So it could be short inserts, or you have reads on either end of the DNA, and once you get the sequences, you are going to go and align with the genomic data that is shown here. For example, that would tell you a given segment of transcript has come from representing exon or intron, and whether it is spliced product or unspliced product, all this information can be obtained. So this is how you do that for that. So you have the raw data, and then you align them to make the contigs or assembly, and then you compare them with the reference sequence that talks about, for example, junction and crossing readouts, for example alternative splicing, right. Exon 1 is joined with exon 3 or exon 2 or exon 4. So by looking at the sequence in the exon boundaries, you will be able to tell how many times, how many different ways the exon is spliced. Or you can talk about fusion events. Sometimes your chromosomes are translocated. As a result, two different gene is fused together, therefore you have what is called as a chimeric transcript, transcripts having different coding regions. Easily you can look into that. Or you can look at even what is called as allele specific expression, meaning you have for all the genes that are present in your autosomes, you have two copies, one representing father, one representing mother. For some of the genes, it could be only the maternal gene copy is expressed, meaning the gene that copy that you derived from mother is expressed, not the gene copy that you got from father. So how would you really do this? So you can sequence it, and there are variants that are present, right? These variants, allele could be different between father and mother. By looking at the sequence of the RNA, you will be able to tell, is there that both the paternal and maternal allele contributed equal number of transcripts, or there is a disparity. You have more of from the maternal one or more transcript from the paternal. If there is a difference, then you can go back and look into the genome organization and see is there anything that affects the way the transcription is being done. So that is another powerful tool which is normally you miss out when you do a microarray or real-time PCR that we discussed earlier. And then you can talk about other, you can talk about count matrix, meaning how many reads you have gotten for a given transcript or gene. That talks about the quantity, the expression difference. So if you have a normal skin versus tumor skin, then not only you talk about different ways spliced or so on, but you can also talk about the differential expression, and then you will be able to come up with that. And then since you are looking at a large number of RNAs at the same time, you can model it. If gene A expression level goes up, what happens to other genes? Is it that there are some genes whose expression also goes up? So you are going to have a large number of samples. So you can do a model, and then you'll be able to tell whether such kind of co-expression patterns you can identify, which help you to predict more networks and so on, right? This is just to give you some example as to how people do. This is RNA-seq data. So these are nothing but these mountains represent the reads, how many reads happened for the region of the DNA. You can 5' end of the gene, 3' end of the gene, exon 1, 2, 3, 4, 5, 6, and this is UTR. You find that the number of reads that you found for each segment. So with this way we are able to map it. Not only that, you can go and talk about, for example, if you go for other methods, for example cDNA library or other methods, normally your analysis power more restricted to the 3' end, which sort of is taken care in case of the RNA-seq. So that's one of the major advantages. I gave you example of chimeric gene. When there is a translocation, it could so happen that two different genes fuse together. By looking at the transcript, now here you can find out such kind of transcript where the splicing took place, such that two different genes now transcript is joined together. So this is a powerful method that you have. The last section that we are going to discuss is how the chromatin or the DNA modifications can alter the way gene functions, right? What are the way the the transcription can be regulated? The cartoon represent here the DNA, and then you have what is called as enhancer sequences, onto which there are some proteins comes and bind, which is called the enhancer activating proteins, and you have of course the transcription factors which bind to, and then it all together as a complex recruits the RNA polymerase for the gene to be transcribed. So now whether a gene is expressed or not expressed depends on whether these transcription factors are present and whether this region of the chromosome is open for the protein to come and bind. It happens because of many factors. One of them could be the changes in the DNA, which is called as methylation, or at the chromatin it could be histone acetylation and so on. And how do you really look at such kind of changes at the global level? So one way people do is to do what is called as chromatin immunoprecipitation. So basically what you have is that you have the chromatin here is shown that has a tail coming out, which can be modified. It could be acetylation, it could be methylation, it could be ubiquitination, it could be simylation, it could be phosphorylation. So if you have an antibody which recognizes acetylated histone or methylated histone or any other modification specific antibody, now the antibody would now come and bind to this region. So now if you can use this as an anchor to pull the DNA that are present close to it, then sequence the DNA, it will tell you which are the regions that are in the chromatin is acetylated or methylated. So this is the way. Or I could use an antibody against any one of the transcription factor. Now, and if I pull using this antibody the DNA that are bound to this transcription factor, it will tell me as to which segment of the genome the transcription has bound to. So, this is a powerful method, and this is what called as chromatin immunoprecipitation. The schematic is shown here. So, what basically you do is that you isolate the chromatin and then cross link the protein to DNA. So, you can use certain chemical moieties which stabilizes what is shown here. So, the DNA and protein is stabilized, and then once you have done that, what you do is you sort of shear the DNA, cut the DNA randomly. Therefore, you have a protein complex in which you have the DNA bound to it. So, there are going to be. Now, if you use an antibody which recognizes a given protein, for example, I am looking at transcription factor A. Now, this is the antibody that binds to transcription factor A. So, it is going to pull wherever transcription factor is there in the protein, is going to pull. If the transcription factor is bound to certain DNA, that DNA also would come along with that, after which I extract the DNA and I sequence it, and with the sequence I can go and map the genome to see which region that sequence represents. Therefore, I can tell this is the region onto which the transcription factor is bound to. So, this is the way people do, and this is one such landmark paper where they have looked at high resolution profiling of histone methylation in the human genome. So, they have used this chromatin immunoprecipitation followed by deep sequencing to understand how it happens. So, basically they have pulled the DNA using chip immunoprecipitation, and then they went and did this NGS. You have small fragments that are derived from the chip DNA, you added the adapters, you pass through the fluids, the device, and then you make clones, you make these clusters, several copies of it, and then went through this sequencer to get the data. So, with that, now we are able to tell, for example, the regions in the genome where the methylation is very high, because you use an antibody now recognize, for example, methylation of the histone. Now, it tells here, for example, this segment of the genome you have several reach. That means in many cells this is the segment onto which your antibody is able to bind, which suggests this is the region the histone is methylated. What is that region? It is an upstream region or downstream to upstream to a particular gene. So, it tells you that this region is methylated and this is expressed or not expressed. You can go and analyze later. And this is going to really help because we are talking about epigenetics. Epigenetics is I may have a normal DNA sequence, but the way my genome functions is modified because the kind of environment I live in, for example, right. So, if I can understand now what are the regions that are of the chromatin that are methylated, it is going to help how the environment modify the genome, its function, without changing the base. So, that is a huge advantage in understanding the function. Some of other sequence shown here, for example, this is an active gene, this is an inactive gene, or a condition in which the gene is inactive, a condition in which the gene is active. You can see that this is the number of reads that are denoted by the line diagram. Here you can see the enhancer region. If you have used, then you can find that the methylation is altered here, more reads come here, likewise the promoter and so on. So, this is the way, depending on what you are looking at, transcription factor and others, you are able to infer as to how that could possibly regulate the expression of the gene. So, that is the power of the next generation sequencing, and as you have seen, they are just these technologies are coming up. Still you have a long way to go, and maybe next 5 years, 10 years, you are going to see changes that you cannot imagine. It's beyond imagination, and it could be anywhere in the city, town. There could be shop that can sequence the genome and may have methods to analyze the genome and predict as to what are the risk factors that you have, or diagnosis can become better, just like what you have for blood test and others. So, with that we end this section of the discussion, that is next generation sequencing. The next classes we are going to discuss about the usage of these sequences to understand how we evolved and how we can analyze the genome sequencing.